Hello, I'm Rachel Babin from the Oncology Network. Welcome to the OJC Meets, sister series of the Oncology Journal Club podcast. In today's episode, Craig Underhill meets Ashray Gunjur to discuss their recently published paper on the microbiome. The paper explored how the microbiome could be the key to identifying individuals who can benefit from combination immunotherapy across various cancers, including rare cancers. Professor Craig Underhill is a medical oncologist practicing in Albury, Wodonga. He is a renowned champion of telehealth and teletrials. Dr Ashray Gunjur is a medical oncologist from Melbourne, currently undertaking a clinical PhD at the Wellcome Sanger Institute in the UK. We hope you enjoy listening. This is Rachel Babin, and this is the OJC Meets.
So it's a great pleasure to welcome a long-time fan of the podcast, Dr Ashray Gunjur, who's a medical oncologist trained in Melbourne, now working in Cambridge doing his PhD in cancer and the microbiome. So fascinating area. Welcome, Ash.
G'day. Yes, it's great to be on. What an honour.
So this sort of fulfills three dreams, Ash. We previously talked about if anyone had an interesting paper that they wanted to put forward, please let us know. So you've done that. It's a blow your own trumpet paper. And it's from Nature Medicine, now one of our favourite clever type journals to have on the show. So it's great to have this paper, Ashray, titled A Gut Microbial Signature for Combination Immune Checkpoint Blockade Across Cancer Types. Before we get into the paper, just tell me, before this paper was published, what's a kind of summary of where we're at with the interaction between microbiome and cancer and responses to immunotherapy, because there's been some intriguing work before.
Yeah, absolutely. I think intriguing is a great word. I think it's a really exciting time to be studying the microbiome, particularly the gut microbiome in cancer, but there's a lot of challenges as well, just with understanding the relationship. I think the field really took off back in early 2018 when really pioneering work out of three groups was published concurrently in the journal Science. So these three groups, one in France and two in the US, all studied cohorts of patients with metastatic melanoma. And they showed that within those cohorts, they could find particular species, particular bugs in the gut microbiome that associated with response to anti-PD-1 therapy. But I think what was really exciting was that all three groups would show that in taking the stool from patients and transplanting it into mice, they could recapitulate those responses. So you could have the stool from a non-responder and give it to a germ-free mouse, and that mouse would not respond to anti-PD-1 therapy. And similarly, the stool from a responder would actually induce a response. So it really was encouraging of this causal relationship and the potential application of microbiome-derived therapeutics. But since then, there have been a lot of studies, and what's been really difficult is to actually deduce what the specific bugs are across geographies, across studies, across cancers that are predictive. Because when we look at the multitude of clinical cohorts that have been published since, there have been really different findings across the different studies. There have been some overlaps, but there have been some inconsistencies. In fact, some studies have shown one bug to be good while another has shown that bug to be bad. So we really wanted to understand the cause for these inconsistencies and see if we could improve the generalizability of the microbiome in being something that might be able to predict patient response, in this case to cancer immunotherapy, or that something we might be able to target to enhance patient response.
Fantastic. And it kind of makes sense biologically, because we know that microbiome is part of the innate immune system and there's crosstalk with the adaptive immune system. People have really kind of leapt ahead though and started talking about giving patients antibiotics or not, or giving them probiotics or faecal microbiome transplants. So there's a lot of kind of work and exploration. So then tell us about your study and what we learned.
Yeah, absolutely. I guess the setting was this kind of challenge in defining really consistent gut microbial signatures. We call them signatures because it's kind of like a pattern of microbes that in this case is predictive of immune checkpoint inhibitor response. So the challenge of finding generalizable signatures of response from the gut microbiome, given those inconsistencies, but knowing that there was promise from those preclinical studies, those mouse studies showing that you could actually improve response. And there started to actually be some human data, fecal microbial transplant, stuff like that. And of course, the retrospective evidence around antibiotics. So one of our hypotheses was that maybe one of the problems, in addition to the problems that had been previously covered about differences between the gut microbiomes across different communities, across different countries, maybe one of the other problems was that actually we weren't looking at a deep enough resolution at what the bugs were. Because conventionally, we look at bugs at the species level or even a higher taxonomic rank, that the genus level. So we lump all bugs together that are part of the same species, and bugs, they're prokaryotes. So we lump species based on a, I guess, generic threshold based on how similar the genomes are. So you might have lots of different bugs that we call the same species that do very different things. And we actually know that from the work on pathogens. So when you've studied E. coli, you know that some strains of E. coli are completely benign, while others can cause horrendous disease. And the difference might just be 1% of the genome. So we thought, well, maybe that's the case for commensal bugs as well. Maybe the ability to have these consistent signatures would be enhanced if we could look down at a subspecies level, a strain level. So that's what we set out to do. So we started off with the great rare cancer study, which you know well, Craig. It was a study run out of 5 different sites across Victoria and New South Wales, including Albury-Wodonga.
That's right. It was one of the first teletrials done in Australia using the teletrial methodology. So, yeah, close to our heart for many reasons. Gave patients in regional Victoria access to immunotherapy at a time when it wasn't readily available on the PBS. So, sorry I interrupted you, but keep going.
No, yeah, absolutely. It was super exciting, and it was also patients with really rare cancers who, are historically underserved, who often lack standard of care treatment because their diseases are just not well studied enough. And it was a basket design, so, various different cancers, cancer types, patients with advanced cancers of the gynecological tract that were rare, neuroendocrine neoplasms, or upper GI or biliary tract neoplasms, were all treated with ipilimumab and nivolumab in a pretty conventional fashion. And actually, amazingly, 25% of patients responded across those 3 pre-specified tumor cohorts, which was just fantastic. And we saw that those responses were durable. So it was a fantastic study, but I think it was especially fantastic just because of the translational research opportunities that were built in. And, full credit to you, the other PIs, and of course, Oliver Klein and Jonathan Sabon, the coordinating PIs who had the foresight of collecting all these samples. So one of the biospecimens that was collected from the majority of patients was a pre-treatment stool sample. So that's what we used to assess the gut microbiome, or in this case, the fecal microbiome. So we extracted the DNA from those samples, and we used something called shotgun metagenomic sequencing. So this is where it's kind of the equivalent of whole genome sequencing of the metagenomes, because there's lots of different species, lots of different genomes in that stool sample. But you have a really deep understanding of the whole genomes of what is present. And then we used some bioinformatics to not only stitch together what the bugs were at the strain level, but then also map the reads to those genomes to really get pretty accurate quantifications of how much of the bug was present in each of those pre-treatment stool samples. And then we could actually use it to predict response or progression across these very, very different cancer types, because we had that information in follow-up. And what we found was when we used the strain level information, we actually saw an improvement in the predictive performance when we cross-validated across this very diverse rare cancers cohort than when we used the more conventional species or genus level information. So I think it, to us, really affirmed that hypothesis that having a higher resolution, higher granularity of the microbiome does actually make an improvement. And it actually allowed us to cross-validate against very different cancer types. So when we trained the model just on 2 of those pre-specified tumor cohorts, we could actually quite reliably predict it on the left out tumor cohort. So this was really exciting to us that, not only could we see an improvement in that cross-validated performance with the strain resolution, but we could actually see generalizability across cancer types. So then we wanted to take it a bit further and look at public data. So there is, fortunately, a lot of published data from predominantly patients with metastatic melanoma. Their pre-treatment gut microbiomes subjected to shotgun metagenomic sequencing, so stool samples, shotgun metagenomics. So similar design, obviously some differences in terms of the specific kits that we use and the sequences that we use that do introduce some batch effects. But we were able to reanalyze that data in the same way. And we found another interesting thing. We found that the signature from the rare cancers trial, where all patients were treated with ipi-nivo, so anti-CTLA-4, anti-PD-1, seemed to only perform well on external cohorts where the patients had received ipi-nivo, so anti-CTLA-4, anti-PD-1. So it didn't seem to perform very well. In fact, it performed like a coin toss when we tested it on patients who just received anti-PD-1 monotherapy. And this was a little bit surprising because we always thought of anti-PD-1 as kind of the workhorse. It's carrying the combination immune checkpoint blockade efficacy. But I guess when we thought more about it, it seemed like there was already evidence that the mechanism of action of combination anti-PD-1, anti-CTLA-4 is quite different, and that the addition of anti-CTLA-4 probably does make a difference, especially in the interaction with the gut microbiome. There's evidence that it can change the permeability of the gut. Obviously, colitis is a much bigger phenomenon with anti-CTLA-4 inhibition. And there was evidence from other sources of data, like immunotranscriptomic data, that the immune responses were quite different to this treatment. So it suggests to us that there is a common signature of response to immune checkpoint blockade across cancer types, but there might be different microbial signatures to different regimens, which we thought was interesting and we wanted to share.
So in summary, I think it's really important information. So the signature of response is predicated on the treatment regimen rather than cancer type, which is the conclusion in the paper. And so it serves an important knowledge base for further trials looking at microbiome diagnostics or any interventions. So is that a valid conclusion, do you think, Ash?
Yeah, absolutely. I think you're exactly right. I think this is a foundation, but maybe future studies could unpick this further and focus on that split in different immune checkpoint blockade regimens, because historically we were sort of lumping them together, right? Anti-PD-1 monotherapy and combo anti-PD-1, anti-CTLA-4. They were both just immune checkpoint blockade. And now obviously we're going to be studying combination IO chemo for a lot of cancer patients. So that's possibly a different kettle of fish as well. So I think that this regimen is a really important covariate. Obviously cancer type is too, but we were actually reassured that there were quite similar signatures across different cancers in terms of what defined or what predicted a future responder versus someone who unfortunately wouldn't respond.
Fantastic. So look, really intriguing. We've had other papers over the years about microbiome. I think it's a really fascinating area. Might be even worth an episode down the track summarising the whole area, but I think intriguing is the word of the day. Let's hope that this knowledge will then lead to tools that we can predict people's response and maybe alter the microbiome if they are unlikely to be responders. Really fascinating area. Ashley made it sound really simple, straightforward, and it's not. Like the technical stuff in the paper is really quite high level. So a fantastic paper. Well done. Congratulations on all the work you're doing.
Well, it was a real team effort, and we couldn't have done it without you and all the sites recruiting to this great trial. So thank you.
Yeah, thank you, Ash. All the best for finishing your PhD, and let's hope we see you back in Australia in the near future.
Yeah, I hope so too. Thanks so much, Craig. Thank you so much for having me on.
You've been listening to the OJC Meets, proudly produced by the Oncology Network. For regular news and podcast updates, we invite healthcare professionals to join us at the Oncology Network website, oncologynetwork.com.au. Your free registration includes a free subscription to our weekly publication, the Oncology newsletter, and it's a fantastic way to support the OJC. This is Rachel Babin, and this is the OJC Meets podcast.